These are a class of pharmabiotics. So as Teresa pointed out, you can only be a probiotic if you're alive. You can be a pharmabiotic even if you've been heat killed. And just because they cannot be probiotics doesn't mean they can't be beneficial. And heat-treated cells, as it says there in the bottom, could have very significant advantages. They can't colonize, they can't infect, they can't die during storage because they're already been inactivated. So there could be significant advantages to using pharmabiotics. And I wanted to start by giving you one example. Or firstly, let me say there's this review which has shown there have been at least 50 studies in humans using heat-inactivated bacteria. Lots of different bacteria, which you can see here, and lots of different health conditions, which you can see over here. And in many of these trials, there were benefits shown by the use of these pharmabiotics. One really nice recent example, this was published in Nature Medicine just last year by Patrice Cani and his group. They showed that this bacterium, Akkermansia mucinifila, can have a beneficial effect in obesity. So if you give this single bacterium to mice on a high-fat diet, they do not put on as much weight and they do not develop metabolic syndrome. A single bacteria with this incredibly beneficial effect in metabolic syndrome. But what they found, much to their surprise, because they did this as a control experiment, they said, let's kill the bacteria and show that a killed bacterium has no effect. But when they did it, it actually turned out that the heat-killed bacterium had a bigger effect than the live bacterium. And that's reported there, as you can see in the title. Pasteurized bacteria improves metabolism in obese and diabetic mice. So an extraordinary example here of a heat-killed bacteria actually working better than a live bacteria. Now, we've been working for the last 2.5 years or so on this particular bacterium or bacterial preparation, Lactobacillus lb. And Lactobacillus lb is a pharmabiotic. It contains 2 strains, you see them here. It's grown up to about that level, to very high levels, and then it's heat inactivated. And of course, the final preparation also includes all of the molecules that it produced during growth. So we don't just have the bacterial biomass, we have the molecules that it made during growth as well. And we started out by sequencing both bacteria and looking at every gene in the bacteria and looking to see that they don't have any harmful genes, they don't have antibiotic resistance, they don't produce toxins and so on, that they're very safe. And then we designed this trial, and this trial at first might seem like a fairly unusual trial. What we wanted to do was see in mice whether if we fed the mice the heat-treated Lactobacillus lb or a control, which didn't include this product, and then we followed these mice for 8 weeks, and in the middle we did a whole series of behavioral tests on the animals. We worked with a psychiatrist, Professor Ted Dinan, and it's very strange for a psychiatrist to be working with bacteria and dead bacteria, but Ted has a very open mind, so he did this work with us. And then we subjected the animals to a series of behavioral tests. A lot of these tests are to do with the stress experienced by the animal. Others are to do with memory and cognition and things like that. We also analyzed the microbiome every week to see, does the heat-killed bacteria have an impact on the microbiome? And I should just do a small bit of background here. I'll bring up these 3. There are 3 ways of studying the microbiome. There's more than 3, but the 3 I'm going to mention are, you can look at the individual microbes within each different sample. So these are different, each one of these is a different sample, and each one of these colors represents a different microbe. You can look at things like beta diversity, where you're looking at each individual is one single point, and the closer 2 points are to each other, the more similar the microbiomes are. Or you can look at diversity, which is a measure of how many bacteria are present and how diverse they are. So you can compare the microbiota looking at very different things. So we looked at the microbiota in these animals getting the Lactobacillus lb, and what we found was that in general, whether you got LB or the control, your microbiome the major species in the microbiome didn't change very much. And this was good. We didn't want it to have a major disruptive effect on the microbiome. But in these smaller, lesser species that you can see here, we could see some differences occurring. Also, we found there was no difference in diversity, which again is good. You don't want to really change the diversity too much by administering a dietary supplement. But when we looked at beta diversity, what we could see is that the microbiome of the mice receiving the Lactobacillus LB changed significantly from the control mice. So at the start of the experiment, of course, they all had the same type of microbiome. By the end of the experiment, the control mice were here and the Lactobacillus LB mice were here. They had shifted. And so this shows us that the Lactobacillus LB is having a profound effect on the microbiome, not by changing the major species or changing the diversity, but by changing some of these minor species. And then we looked at the impact on the brain and on behavior. And much to our surprise, we saw very significant changes in behavior in these animals. So for example, here, these are different tests. In 5 of the 8 tests, there was no difference. But in 3 of the 8 tests, there was a significant difference. Let me just show you one of them. Here's the tail suspension test. It's a horrible test. I didn't make it up, and I don't like doing it. But what you do is you tie a mouse by its tail to a kind of a wire like this, and you just hang the mouse upside down. And the mouse will struggle to try and get upright again. But you can measure the amount of time the mouse spends struggling and the amount of time the mouse just simply relaxes. And it's a measure of how stressed the animal is. And this Lactobacillus LB had a very significant calming effect on the mouse. And these 2 other tests as well, it showed a calming effect, an anxiolytic effect. So the product had a calming effect on these animals. And when we looked at corticosterone, which is the main stress hormone that indicates anxiety, we found the same thing. The control mice had normal levels of corticosterone, but the mice that were fed the Lactobacillus LB had much lower levels of baseline corticosterone, showing that they were in a much more relaxed state. If we agitated the mice, if we subjected them to a test, then the cortisone, of course, goes up, and it went up equally in both groups of mice. So it doesn't interrupt their ability to react to a stress, it just makes them more calm to begin with, which is a very surprising outcome. And we are starting to understand the mechanism, because what we found was that when we used this product, this Lactobacillus LB, we saw changes in very specific bacteria. For example, Odoribacter were significantly decreased, and the literature tells us that Odoribacter are associated with anxiety-like behaviors. So if you have high Odoribacter, you have anxious behavior, and LB reduces the Odoribacter. It also reduces allostepes, and these have been observed to be very high in patients with major depression. So it's possible that Lactobacillus LB, through the microbiome, is having an impact on the brain and on behavior. But more work needs to be done, and of course, particularly work has to be done now in humans to validate this. And this has been published just recently, about a month ago. So if anyone's interested in reading this paper, you can see the title there, Heat Kill Lactobacilli Affect Both Microbe Composition and Behavior. We've also done an experiment to see whether Lactobacillus LB could impact on inflammation. So this is another experiment where we gave the mice either the Lactobacillus LB or just normal chow. One group of mice just simply got the Lactobacillus LB or the normal chow. The other was infected with Citrobacter. And we weren't looking to see if Lactobacillus LB could prevent Citrobacter infection, because we didn't expect that it would. This is just a localized infection. But what's interesting is that the mice who get infected will almost always go on to develop colitis. And so what we wanted to see was, could we have an effect on the development of colitis by giving the mice LB? And we took lots and lots of samples, and the analysis isn't complete yet. I'm giving you results that haven't been published yet. They're just really recent in the lab. And what we were able to show was the same effect on the microbiome. So we still saw that separation. The mice receiving Lactobacillus LB have a different microbiome by the end of the experiment than the mice who didn't get Lactobacillus LB. We saw that both groups of mice were infected with the Citrobacter, so that was what we expected. But what we were really interested in was the effect on the inflammation and on the colitis. And what we were able to show was, again, this is very preliminary data. I'm not showing you all the data. What we're able to show is that in colitis, the length of the small intestine is reduced in animals who have severe inflammation. That was not as severe in the animals that got Lactobacillus LB, and also crypt depth, which is a sign of a healthy gut, was reduced in those animals that got infected, but not reduced in those animals that got Lactobacillus LB. And now we're doing all the follow-up data to see what the actual mechanism is here by which Lactobacillus LB prevents colitis in these animals. And one clue, one idea that we're looking at right now, this is a fecal fermentation. So this is an artificial gut that we have in the lab. This is Glenn Gibson. You don't actually wear it and walk around with it like this. It's on a lab bench. But what you can do is you can mimic the human gut. And we added Lactobacillus LB into that human gut. And what we saw was a very strong bifidogenic effect. We saw an increase in the level of bifidobacteria within the colonic model, which we don't see in the absence of the Lactobacillus LB. And so it may well be, again, that the impact on colitis is through